Thank you. So we are going to move on to the second speaker this morning, Dr. Byungkook Lim, who is an associate professor of neurobiology in the Division of Biological Sciences at the University of California, San Diego. Byungkook is also a collaborator of my laboratory and working on various projects. And his talk today is Neural Circuits Mechanisms of Early Life Stress-Induced Maladaptive Feeding Behavior.
Thank you for a nice introduction, and thank you for the organizer, Picower Foundation and the Li Hui, for inviting me to give a talk. So this is very early time for me, so I'm a little jittery right now, so we'll see how it goes. So, you know, our brain can be out of order, right? You know that. So let's say we see somebody like this, right? Then, of course, we want to help him, but we really have to know what is going on in his brain, right? As a circuit neuroscientist, I really want to dissect every kind of part of piece of his brain, right? So let's say we know what is going on. Let's say we know the problem. Let's say we fix this. So hopefully we can make him better, right? So that's the goal of many of our research. But finding out the problem in the brain is very challenging. So this is actually the brain scan image of the two patients with the major depressive disorder. So they have a similar age, and then their symptoms are very similar, and they have a very similar history of the medication. But as you can see, their pattern of the brain activities are totally different, right? So this is very puzzling because we really cannot see the brain of the inside of the head of the patient. In fact, a very well-known psychiatrist, I didn't say this, but this Dr. Amen said that psychiatrists are the only medical specialists that never look at the organ they treated. No offense, I didn't say this. But the thing is, we don't really know what is going on because we want to know what is going on, right? So, of course, this difference, this individual variation or individual difference may be coming from the different genetic background, different living environment, and of course, it's different childhood experience, right? So we are focusing on early life experience. So we remember something happened, right, when we are very young. I vividly remember some event, especially a very aversive event when I was 6 years old and 5 years old, even though I don't remember what happened 2 days ago, right? But in addition to those memory, some life experience happened in early life actually has a long-term effect on the mental health. We already heard a lot about ACE, right? Indeed, there's a lot of studies shows that aversive childhood experience is somehow associated with the many forms of the physiological and the mental disorder, right? But question is, how? How would this early life event, early life aversive event, is actually associated with the many of this mental disorder and physiological disorder? What is the mechanism, right? And then also, it also depends on what type of stress, when it happened, how severe this stress is. It's all very complicated process going on, but we don't really know the mechanism underlying those change. So I'm a circuit neuroscientist with the animal model. So there are a lot of tools and idea and a little complicated model system. So hopefully it's, I can convey our information to you a little better. We'll see. So based on this study, actually, we thought that understanding mechanism underlying early life stress is actually crucial to treat the many mental disorder. But we have to get some idea, right? Where are we going to start from? So we look at a lot of literature human literature to see what is actually the very important correlation between early life stress and some physiological disorder. So there is many study really shows that the childhood childhood adversity is actually show the significant association with obesity and the binge eating behaviors. And of course, a lot of other substance disorder and stuff like that. And as you can see, the people who experienced strong trauma in the World War II or some other terrible time also reported the high rate of the binge behavior. So it was interesting, right? How those stress, early life stress, has a strong association with the feeding behaviors. More fascinating thing, it's a terrible story, but we're very interested in this story. So this report shows that the children who went through this terrible time, especially when they're very young, like this story shows that children who went through this time at the age of 2 or 3 still show the higher correlation with the binge eating behavior and obesity. Think about it. Year of 2, 3. You don't really remember what happened at that age, right? But still, that stress event actually has a long-lasting effect on their physiological, behaviors. So we actually start working on this. What happened? How? The stress at the really young age has any impact on long-term, physiological behaviors. Like I said, we are working on Loden. So based on this idea, we actually developed this behavior paradigm to give a very strong stress at the early time. So as you can see, we actually separate pup from mom and sibling. So it's like a total social isolation for 23 hours, basically for 1 day, right after right after birth, like P3, right? So this basically 1 day period, they don't have access to the mom, they don't have access to the food, and they don't have any social interaction with others, right? That's it, just 1 day strong, stress, and then this pup go back and then actually develop normal way. And we actually looking at their developmental trajectory, their body weight. Actually, at the really early stage, their body weight is a little reduced compared to the control group, but they catch up. So actually, when we are actually looking at the behavior and all the physiological measurement, actually, they don't show any difference in terms of the body weight between control group and then animal exposed to the early life traumatic event. We call this ELT, early life trauma mouse, right? In addition to the body weight, actually, we look at the stress hormone level. Of course, as you can see, the P4 right after the stress, there's a strong increase in the corticosterone, stress hormone. But when we are actually doing some behavior measurement, there's no change in the stress hormone level compared to the control group. And their actual movement is totally fine. This is the measurement for the anxiety. Basically, they don't show any anxiety phenotype. And we actually measure the social behavior and others. Actually, they have a very normal social behavior. So they look normal, right? Just 1 day of a very strong, traumatic event. Yes, brain is plastic, right? They can kind of make things better. So looks like this mice is normal. But when actually we introduced high-fat diet, basically this is the kind of the high-fat diet we are using. This is a normal chow, basically the lab chow every mouse used. And then actually the high-fat diet, actually total calorie is the same, but we actually increased the fat content significantly, right? They love it. Anyway, so when we actually give the normal chow for a long time, as you can see, they look normal. Their body weight gain and then actually total body weight is actually same as control group, right? But when we introduce the high-fat diet, basically they can choose the normal diet and high-fat diet at the same time, but it's just dump the high-fat diet there. as you can see, their body weight gain is significantly increased, and actually their body weight go higher, right? Somehow, they look normal in many behavior tests. They show significant difference when we actually put the high-fat diet. So this is interesting, right? So first we thought that, okay, maybe this ELT mice exposed to the early life traumatic event, may have a different body metabolism. That's why actually they process the high-fat diet differently. So we actually tested many physiological tests. First one is actually inject the glucose, see whether they actually process glucose same way as a control group. So there actually the metabolism in terms of processing the glucose looks very same. And we also measure the overall metabolic activity. So basically we measure the oxygen consumption and CO2 exhaustion, and look at the activity during the day and during the night. We didn't see any change. So it looks like those short-term stress may not have a significant impact on their the metabolism. And then it's probably something going on in the brain, right? So as I mentioned to you in the human study, early life stress is actually associated with the increased binge eating behavior. So we thought that this increase in the body weight in the ELT mice may be due to the increased binge eating behavior in this animal. So we actually look at the literature, and then we actually developed this some interesting paradigm. As we call the binge eating cycle paradigm. So here it's a normal control group and ELT group. They never exposed to the high-fat diet until we actually introduced this high-fat diet. So we introduced high-fat diet for 2 days. So they know some good thing is out there, right? This is the first time they encountered the high-fat diet. And then actually we remove the high-fat diet and then go back to the normal chow for 5 days. So good things gone, right? And then suddenly we introduce the high-fat diet just for 1 day. And then they really love it, of course, right? Good things came back, right? And then remove the high-fat diet again, and then actually go back to the normal chow for 5 days. And then go back to the high-fat diet 1 day, and then go back to the normal chow for 5 days. It's actually do the cycle. So first time when they are encountered the high-fat diet, actually they love high-fat diet, but they don't show much change, right? But when we do this kind of cycle, yes, at the first, just by looking at the control, they show the strong binge eating phenotype, basically the same duration, 2.5 hours. But usually when we had introduced the first time, actually the mouse take up little higher than 0.5 gram per day, per 2.5 hour per animal. But actually, when you do this kind of cycle, they eat a lot. But when you look at the control group, they eat way more. And then, ELT group, we eat way more. And the control group kind of tend to slow down their trend. But actually, the mice exposed to the ELT actually do increase their tendency of getting a high-fat diet. So they show the augmented and then actually sustained binge eating phenotype when they're exposed to the early life traumatic event. And the eating rate is way faster compared to the control group. So, okay, why they have this augmented and sustained binge eating phenotype, right? Basically, this data kind of prove that the early life traumatic event can actually generate the binge eating phenotype, right? In human study has a lot of correlation, but using some animal model, we show the possibility. We actually show some causality here. But we want to know the mechanism, right? So we actually do the very simple activity screening called CFOS staining, right? CFOS is an activity marker. You can do immunostaining, basically detecting the protein in the whole brain and see which brain area is activated, right? So we found this kind of brain area. So control area group, without the high-fat diet, there's not much change in the CFOS staining. But when we first introduced, yes, there is a strong increase in the control group and the ELT group. But when we actually keep doing binge eating cycle, the last cycle, the fourth cycle, actually control group actually reduced the CFOS, which was increased in the first encounter, first cycle. But the ELT group actually showed the sustained activity as measured by CFOS. So we thought that this sustained CFOS activity, basically neuronal activity in this area, may be responsible for the sustained binge eating behavior in the ELT group. So this brain area is actually the lateral hypothalamus. So, lateral hypothalamus is very important brain area, but it's a very complicated brain area. Lateral hypothalamus is involved in many physiological functions, including feeding behavior, sleep and wake cycle, and energy balance, and kind of everything that you need to live. So, this is interesting, kind of makes sense, but when you actually study this kind of complicated brain area, you really need to know what type of neuron, what type of molecular signaling is involved in this process, because it may do many different things, right? So to understand, to actually examine what actually the molecular pathway or neuronal cell type is responsible for this early life stress-induced binge eating behaviors, we actually perform the quantitative RT-PCR against the many neuromodulatory signals. So what we found is, surprisingly, the leptin receptor mRNA is significantly reduced in the lateral hypothalamus in the animal exposed to the early life stress. Leptin receptor. Leptin, is a very important hormonal pathway. This leptin is secreted from the fat cell, and then actually this signaling going to the brain actually has an important role to suppress the food intake, right? If you eat too much, or if you're obese, actually this signaling going up and suppress the food intake. So if you have an issue with this signaling pathway, basically you become like this. So this is the mutant mice, actually the OBOB mice that has a mutation in the leptin receptor signaling. And leptin receptor is actually expressed in the many areas in the hypothalamus, including lateral hypothalamus and the arcunucleus hypothalamus too. So looks like leptin receptor signaling in the lateral hypothalamus is reduced in the animal exposed to the early life traumatic event. The question is whether this reduction of the leptin receptor is actually inducing the binge eating behaviors. To answer that, we actually developed the adeno-associated virus expressing shRNA against the leptin receptor. So shRNA is bind to the mRNA of the leptin receptor, so actually reduce the leptin receptor expression in this specific area. As you can see, the priming, that when they are first encountered the high-fat diet, we don't see much change, but this leptin receptor, reduction of the leptin receptor in the lateral hypothalamus alone, without any early life stress, we can actually mimic the effect of the early life stress. So they're able to show very strong and augmented and sustained binge eating phenotype when we just knocking down the leptin receptor in the lateral hypothalamus. So looks like, knocking down of the lateral, leptin receptor signaling in the lateral hypothalamus may be, enough to induce the binge eating behavior in the ELT animal. And then how they are controlling the leptin receptor, expressing neural activity, right, in the ELT mice. So to directly monitor the leptin receptor expressing neuron in the lateral hypothalamus, we perform some in vivo imaging experiment. So we actually get the leptin receptor Cree animal, which expressing Cree recombinase in leptin receptor expressing cell. And we can deliver the genetically encoded calcium indicator in this specific neuron. And then actually put the green lens and the miniscope and to monitor the activity of this neuron in freely moving animal, right? And then we introduce the normal food and high-fat diet and stuff like that in freely moving animal. So this is one of the I guess one of the best way to monitor the neural activity in response to the food in real time and in vivo. hold on. So, and then we measure the activity at the specific time, and then we can actually measure the activity with the same animal, which is actually better, right? So what we found is when we introduce the normal chow, this neuron actually doesn't show much, activation. We didn't do any like food restriction or anything, just like normal condition. When we introduce the normal chow, even though they approach to the normal food, actually they don't have a strong activation. But when they are approached to the high-fat diet, they show the strong activation in control group. And of course, yes, mice exposed to the early traumatic event is showing the same pattern in the first cycle. But remember, we keep doing like a multiple cycle. We look at the activity at the first cycle. And control group, the activity in response to high-fat diet is significantly reduced as compared to the first cycle, right? But activity of this neuron in the ELT mice still has a strong activation. So it's kind of consistent with the CFOS staining data we shows, right? So somehow in ELT animal, the leptin receptor expressing neuron in lateral hypothalamus shows sustained activity in response to the high-fat diet. So it looks like ELT animal, there is a reduction in the leptin receptor signaling, but also the increase in the leptin receptor expressing neuron activity. Then whether this activity is really important for the generating the, this binge eating phenotype, right? Augmented and sustained the binge eating phenotype. So I'm a circuit neuroscientist. To fully understand the functional role of this neuron, we really have to know where they are connected to, right? We need to know the connection. Brain is network. So first thing we did, it's difficult to see, we try to see where they're making a connection and where they're making a synapse onto. So we found that there's two major areas that these neurons are making a synapse. It's a ventrolateral periaqueductal gray and also the medial preoptic area. Interestingly, these two areas also involved in the feeding behaviors, right? Medial preoptic area actually regulate many physiological function, and the periaqueductal gray is actually involved in many motivational behaviors. And then we found a little more careful anatomical dissection shows that this leptin receptor expressing neuron lateral hypothalamus actually project two different targets, but the neuron projecting to two different targets are actually the separate population. So using some virus tracing method, I'm not going to talk too detail. So we are dealing with the two different population and the two different projection. The question is, which projection is more involved in this binge eating behavior, right? So we actually do in a lot of analysis, but here I show you the pathway-specific chemogenetic manipulation, right? Chemogenetic manipulation is basically the injecting some specific ligand, you'll be able to control the neural activity. So when we introduce HM3D, which is activating neural, increasing the neural activity, but this purple one HM4D is actually decreasing the neural activity. As you can see, when we activate this specific population, we are able to increase the binge eating behavior. It's kind of consistent with the leptin receptor activation. But when we suppress this neural activity, it's actually we are able to suppress the binge eating behavior in this animal. But when we actually manipulate this projection to the medial preoptic area, the manipulation of this neural activity doesn't show any change. This strongly suggests that leptin receptor expressing neuron in lateral hypothalamus projecting to the ventrolateral periaqueductal gray is actually responsible for the binge eating behavior that you see in the early life stressed animal. So we actually show that the early life stress induced binge eating behavior is mainly due to the activation of this specific pathway. Then actually, we see whether we can block the early life stress-induced obesity by reducing this neural pathway activity. to chronically, reduce the activity pattern of this neuron, using some virus tool, we selectively express a potassium channel, which suppress the neuron activity in the neuronal population projecting to the ventrolateral PEG. And actually, we performed the surgery in the mice exposed to the ELT. As you can see, as I showed you previously, the mice showing the ELT has a very increased body weight gain, right, over time and become obese. But by selectively actually inhibiting this neuron activity, we are able to actually rescue the phenotype. Basically, we are able to block the early life stress-induced obesity behaviors. So I think this is kind of all I have. When you're thinking about the circuit, of course, early life stress may have an impact on many circuits in the brain, but some circuit may be more preferentially affecting the specific behavior, right? So we shows that early life traumatic experience lead to, indeed, the high-fat diet binge eating behavior, as you see in many human literature. And we found that there is a specific reduction of the leptin receptor expression in the lateral hypothalamus. And then, we showed that, basically this neuron expressing leptin receptor in the lateral hypothalamus project many different targets, but this specific projection, which projecting to ventrolateral PEG, is actually to have a long-lasting change, and by selectively manipulating this pathway, we can actually rescue the early life trauma-induced maladaptive binge eating behaviors. So we shows that yes, early life trauma, at least in the animal model, we kind of confirm and then verify that early life traumatic event can induce maladaptive feeding behavior, inducing obesity and binge eating behaviors. But brains are networked, right? It's like connected to each other in many areas. So I have to say that we probably find one of the many important circuitry underlying this ELT, early life stress-induced maladaptive feeding behavior. So, there's a long way to go, because we really want to see what is going on and then try to find out all the map. So we need more thing to do to dissect out the more circuitry, dissect out the more dot important for the specific behavior to fully understand the early life stress-induced maladaptive, behaviors leading to the many mental disorders. So, thank you. Hopefully, I didn't went through too fast. Why eating? Yeah. Yes, actually, it could be some other stuff, right? There's a strong correlation between early life stress and substance disorder, and there are some other, paper we published that a little bit of elongated and extensive early life stress also has an impact on social behavior too. So I think timing and the type of early life stress also important, but we screened out many things, and then actually we saw the strongest phenotype in the feeding behaviors. But I would say that if you study some other substance disorder, substance use, substance disorder and stuff, I think there will be some strong correlation with this early life traumatic too. So we found out, but one at a time. We probably have to do more to get the relationship between other, phenotype too. Yeah. Yeah.
Do you have a sense or how you would look at, how does just one day cause such a lasting change? Is the circuit happening to be forming at day 3, and if you don't get it right on day 3 in the mice, it's just like that forever?
That's actually very.
Question.
Yeah, that's the last one.
Can you repeat the question for the virtual audience? Yeah.
I see. The question is, how does the kind of very short-term and early life stress has this kind of long-lasting, circuit change, right? So that's actually our questions too, because at this early age, there is no clear development in the HPA axis, right? HPA axis come a little later than actually the P3, of course, like P5 or 6. So it's possible that they may have a different stress responsive signaling pathway, which we don't really know what is going on. So we really need to do some developmental study to show that, but we haven't done that yet because our focus was actually connecting the early life and then adult behavior. But yes, we have a lot of huge gap to fill out. But thank you for the nice question.